All right, let's get started. So, good evening and
Do we
welcome
make
to
people
everyone.
introduce their animals?
I'm sorry?
I see another animal that hasn't been introduced.
I know
Oh,
there's a dog, and then Mike has a dog too. There's Whistler. Oh,
Oh,
and Kevin has a dog probably too, and Michelle has some cats and dogs. So that we
look
all
at all of them.
look at.
I love it.
All right. Look at that dog, Kevin. All right, so welcome to our Thursdays at 7 Zoom calls. Tonight's call is brought to you by our wonderful friends at Grifols. My name is Madonna McGuire Smith, and I'm the Executive Director of the Hemophilia Foundation of Oregon. We're now changing our name to the Pacific Northwest Bleeding Disorders. So you'll soon see PNWBD showing up in places. We're delighted you're joining us. To get started, here's a few logistical things. First, you're welcome to have your video on or off. We will be recording and we will have it available on our website. So if you don't want your face to show on our recording, make sure you turn off your video. I might suggest that you also change your name on the screen to just your first name so that in the recording, I mean, now that it's already here, but in the recording, it might be better to show. So the way that you can do that is in the upper part of your picture, there's 3 little dots and you can rename. And I might suggest that you drop that down to just your first name. And then the next thing is it would be best if everybody muted themselves. So when the speaker is speaking, she doesn't get talked over or accidentally background noise. You can always unmute yourself if you have a question, and you can wave at her if you have a question, and we can call on you, or you can put it in the chat box. So we'll make sure that you get those questions answered. From time to time, we're going to share our home screen. And I'm going to start out by sharing with you our Thursdays at 7 educational programs and what we have coming up. So next week is going to be outstanding. We're going to have a virtual campfire, and people like Badger and Crooked Branch and Big Dog and Bibli and Garma and Yeti and all of our fun friends are going to join us next week. And we're going to sing a lot of camp songs. So families, if you haven't been involved in camp before and you want to see how it works, join us next week. Wear your camp shirt. And we'll hopefully have some surprise guests with us next week as well. The other thing I wanted to show you was, and I don't know how to do this. I'm not that savvy yet, so we'll figure this out. I wanted to show you that I can never find it when I need it. There are also some information about our Tuesdays at 2. Here we go. Can you see it yet? Not yet. Okay, we'll figure this out. Right here we go. No, that's not what I wanted to share yet. Oops. Okay, we're going to get this. Now it disappeared on me. Anyway, here we go. Tuesdays at 2. It's our lineup for Get Moving on Tuesdays at 2. It's really a fun workout with our physical therapist. And this coming Tuesday, you have to kind of build yourself some things in order to be ready for next Tuesday at 2. And you can find out how to build those on some videos that are on our website. So if you go to our website and you select the link for our Tuesdays at 2, you'll see the video that shows you how to prepare for Tuesday, this coming up Tuesday. And the other thing I wanted to share with you is if you have any questions about how to get in touch with HFO or how to get in touch with me or find our website, there's some information about us. Take a picture of it if you haven't already. But I suggest that you keep that in your little back pocket so that at some point if you have questions, you can contact us. And as I said before, it's a recorded video. But the other really cool thing we've been doing is we've been recognizing seniors. They're not going to get to go through graduation. So I started to show this earlier, but I want to show our 2 seniors tonight. We're recognizing Jorge Castillo. Jorge is part of the class of 2020, and he is from Southern Oregon. And the other one is, not going to show, let me show you. You get to see Jorge twice. All right, let me find the other one is, wow, it's just not letting me do it. Hold on. We might have to show the other one at the end. Let me find it. Here we go. Is she showing it? No.
No.
Share my screen. Sorry, I'm not so good at this yet. So here's the other one. Lucy Serba. And Lucy is graduating in the Portland area. So we're happy to be able to recognize our seniors as we have this awful world of COVID this year. All right, so let's get started. So the first thing we're going to do is we're going to put up a polling question, and I want you to be able to answer this question on your screen. All right. How does Grifols screen for COVID-19? You can select your answer. Got a lot of responses coming in, looking good. Okay, here we go. Here are the results. Looks like everyone was right.
Everybody got it right. Yeah.
Okay, so at this point, we are going to turn it over to Colleen Fraser from Grifols, and she's going to take it away.
Thank you, Madonna. First of all, Madonna, I want to tell you thank you for making me feel so welcome in this new position. You know, I've only started last year, and you really just welcomed me with open arms and embraced me and taught me a lot about the community. And I feel so blessed to be able to work in this community.
Oh, good.
So thank you for that. And then I want to thank everyone that logged on for coming on this evening to see the presentation on plasma safety. I think it's very timely, and hopefully it'll answer questions that can help you, you know, going forward. And without further ado, I'm going to go ahead and introduce our speaker, Virginia Kraus. Virginia is not only a clinical nurse, but she's also the Senior Clinical Education Manager for Grifols and works with the bleeding disorder community. Virginia brings with her over 40 years of experience, and I will go ahead and turn it over to her. But before we do, Madonna, do you want to go ahead and put up that second polling question?
All right, here it comes. Okay, there's 9 out of 10. There's your results.
I didn't see them come up, did they? Oh, there
There
it
we
is.
go.
Okay.
Okay, should I go ahead and share my screen, Madonna?
Sure.
Okay. Okay.
Here we go. Well, thank you for having me, everybody. I'm delighted to be here. I'm actually, this always amazes me. I'm talking from Massachusetts, where we started the day out today at 29 degrees. But we're going to talk about the COVID virus to start out. I don't have slides that address that, but that's obviously the thing that we're most focused on. But when we talk about plasma safety, and we're talking about plasma collection, both from the donor side and from the people who work in the donor centers, we need to look at the safety involved for both of them, for both the donor and the people that work there. So the screening question that you had at the very first is what happens each and every time somebody comes to work. They go through that particular screening, and each and every donor that comes goes through the screening again. So we're looking at whether they have a fever, whether they have a sore throat, whether they have shortness of breath, whether they have any of those outward signs, fever being the most obvious. And then we will further look at what type of exposure they've had. The donors have all been notified in advance about the warning signs for them to be looking at. And they also carry a letter because they are essential people. When we talk about giving plasma, we're not just talking about, and we'll go to the next slide if you would, Colleen. We're not just talking about just the making factor. So we're looking at making those collections to take that plasma that gets donated and pull out all of the therapeutic proteins that we use to make all of our different medicines. So when we do that, that term that you'll hear used is fractionation. And if this refers to the first question, to fractionate out and make those proteins can take anywhere from 10 to 12 months. So we are fractionating out and pulling out all those therapeutic proteins. The very first thing that we pull out is factor VIII and von Willebrand factor. And then we have what we call cryopoor factor 1, or fractionation 1. And then the next one we pull out is factor 9. Our fraction 2+3 is where we make immune globulins for people with immune deficiencies or autoimmune disease. Our fraction 4 is where we make our hyperimmunes, where we make a product called prolastin, which is for genetic emphysema. So these are people that are developing all of the COPD type chronic lung conditions in their 30s and 40s. Again, it's a genetic disorder. We pull out our, with fraction 4, we also pull out the proteins that make our thrombate for people that have issues related to clotting. And then the very last fraction that we pull out is albumin. And albumin is fraction 5. Albumin is used in critical care settings. And obviously, when we're pulling out all these proteins, our whole goal is to make these products as safe as they can possibly be. So we're going to talk about that tonight. We're going to talk about the collection process, the testing that each and every unit that gets donated goes through. We'll talk about the manufacturing process where we're doing this fractionation, and then we'll talk about the formulation. Next slide. So what is plasma? Plasma is, again, when we look at this little vial, plasma is the yellow substance that you see at the top there. It's actually 55% of our total blood volume. There's a lot of water in there. And this is where our proteins travel into our body, and we are fueling our body through the use of plasma, moving our red blood cells. So again, many proteins contained. And for tonight's purposes, we'll talk about factor VIII and factor 9 and von Willebrand factor. And if you remember, as I said, the first fraction that we use is fraction 1, and that's where we get our clotting factors. Okay, so how do we collect it? The process that we use for collecting plasma is called plasmapheresis. And this is done in a dedicated center where the only thing they do in there is plasmapheresis. And that's called source plasma. So people have appointments, they go and they go to the center, and they're actually there to donate plasma. We do sometimes also get plasma from blood centers that have collected whole blood, and they're not actually going to use the whole blood cells, and they'll spin it off and take the plasma, and that's called recovered plasma. But tonight we're talking about plasmapheresis at a plasma center. So again, what's happening here is that the person is actually having a process where they have a large needle placed in their arm, and the machine is loaded with a disposable set. And what it does is it pulls off the red blood cells in the plasma, yet it returns the red blood cells. So somebody that's donating plasma can donate more often because we aren't taking those red blood cells that they need, and the body's able to regenerate and repurpose itself with plasma within 72 hours. So the donation time itself takes about 90 minutes. And because our body can regenerate that plasma, people can donate up to twice a week, which is different again from blood donation, which takes about 40 minutes. But again, because we're taking the red cells there, the body takes a while to replenish the red cells. So that can only happen once in every 8 weeks. Next. So our requirements, again, that when we do that, these plasma centers are very strictly regulated, and they have to meet all sorts of federal guidelines and requirements. We also, because we're a global company, we meet the requirements of the different countries that we are providing products to. And in this monitoring, we do what we call the FDA, the US Food and Drug Administration, again, has very strict requirements. We have to meet all of those regulations to be able to license, and they regularly come in and do spot checks of all the donor centers. We have to meet industry standards, which are handled through the Plasma Protein Therapeutics Organization. And Grifols has, again, has standards that they meet that aren't, that don't fall within the federal requirements. They are, in fact, over and above those. So we're looking at those quality standards of excellence. Again, we look at all of if the little bubbles on the side, if you look at those, all of those are different standards that are required by Grifols to make sure that we have good internal quality control, compliance programs, and we are constantly doing audits and checks within that whole Grifols plasma operations. Next. So, besides meeting those, as I said, those standards, we also have our own safety standards that we are meeting. We also, within the communities that we work and have our centers, and we have well over 250 centers throughout the country. This is just in the United States. We have centers in other countries too. But so we meet all of the community health standards. And then to actually be a donor within one of these centers, you have to come in and pass a test, which is different than when we see somebody donating blood. So when you donate blood, you come in, you answer a questionnaire, they take samples and the blood gets tested. You give your blood and you're gone. What happens with a donor of plasma is they come in, they register, they have to show, and I'll show you on another side, proof of identity, proof of residency, and then they also answer the same questionnaire that we see with blood donation, but then they go to a medical screening. We have medical personnel on site where they go through a screening where we're looking at the health of the donor. So we're looking at their blood counts, their liver function tests, and again, I'll review that on another slide. But again, they do that, and then they go in and donate the plasma. As they take about a quart of plasma, so about 900 cc's is the size of a quart of milk, essentially. They pull off an amount of that donation to send off for testing, and then they put it in the freezer within a half an hour of donation. And then you see the term there, qualified donor. In order to qualify as a donor, what happens is that once they make that donation and they send off the test, that person has to come back. If the test is negative, that person comes back to donate again and go through the whole screening a second time. And because what's happened is when they donate, it goes off into inventory hold, which is part of the safety process, because we're testing to make sure that that health of the donor has stayed the same, and it stays in inventory hold for a minimum of 60 days. So again, we're seeing repeated donations and no change in health of the donor. So give you a good example, if that person donated and they didn't like the experience and they only donated once, and if and everything came back negative, it would never get used in making these plasma proteins, because again, it's missing that safety step of that inventory hold, because again, we're monitoring the health of the donor, which gives us the opportunity, if the health of the donor should change, we could go and pull the previous donations. So it's another safety step. The testing that we do, no, not yet.
Not yet.
Not yet. The testing that we do is nucleic acid testing. Again, we're testing for HIV, hepatitis B, C, A, and parvo B19. Now, yeah, so again, this is a good example of all the regulatory agencies that we have to meet standards for throughout the world. So again, in addition to the FDA, which you see up on the right-hand side, we have the French Regulatory Agency, we have European Medicines Agency. All of those, again, have very rigid standards because they're using our products, we meet those standards. So again, we're meeting not only the US regulatory standards, but as well as global standards. Next. So this doesn't have all 250 dots on here, but it gives you an idea of the fact that we are all over the United States with these 250 centers. We have the most collection centers of any of the companies that are based in the United States or in the world, actually. And what it's also showing you is that our testing centers are the two little blue dots in Texas in the center of the state, because each of the donations gets tested. And every time somebody makes a donation, it gets tested. And then when we put it into a pool, the pool gets tested. So all of that is performed in these two very large laboratories in Texas. And if you look on the left down in the LA area, this is where we have our inventory hold. We have a center there, and we have one all the way over into Clayton in North Carolina. And we also do fractionation in both places, in Clayton and in LA, and we also do fractionation in Barcelona. We also have, if you can look in Colorado, there's a center where we train our plasma center staff. We have over 1,400 actually employees within our plasma collection and manufacturing system. Next. So the process now. Remember now, if you're a new donor and you're coming in, you come by appointment. And I'm going to talk a little bit about what we're doing now with the COVID virus. Again, we know that COVID is not a blood-borne virus. It's more of a respiratory virus. However, we're looking at protecting the health of both the donor and the people who work in the facility. So what we do with each person as they come into work each day is make sure that we test them to make sure that they are healthy. And we have internal, actually a group of 6 nurses who monitor and take care of all the employees within that system. And we have now instituted some changes within the centers themselves as far as personal protection equipment and the cleaning that we're doing. So we schedule donors to come in, and there used to be a large waiting area where they would wait in a group. And now that waiting area has been limited to, again, social distancing, very few people. Our donors are requested to wear masks when they come in. Very few people can wait inside the center, and they discourage them from waiting outside the center too. When they come in for registration, a lot of them they can do at kiosks. The kiosk is cleaned between each and every donor. And then they register with a person behind the desk, as you see over there on the left. That person is wearing personal protection equipment, as well as the donor checking in. They have to present their proof of residency, and they're doing that live rather than the other questions and the questionnaires they do on the kiosk. So the registration happens. Then they go in with a medical professional, be it a nurse or EMT or a physician. They go in for a medical assessment. So when they are going through that process, they are one-on-one within a room, and both people are wearing personal protection equipment. Then they go into the donation room, as you can see on the far right. This is obviously before COVID. Nobody has masks on in this one, but the phlebotomist is wearing, you know, a shield as well as a mask, as well as gloves and protection, a gown, protection gown. And the donors are seated every other seat. So we don't have as many donors in there as we always did. And if in fact they are, the seats are facing one head to head, they'll rotate them so there's nobody across from you. So they stagger them again with the every other seat being vacant. So those are some of the things that we've done to make sure that everybody is safe. And then every evening, now they're cleaning the equipment between each and every donor, but every evening it gets a really deep cleaning. So we are constantly monitoring for making sure that things are kept safe, all surfaces are cleaned. And again, we're protecting both the employees as well as the donors. Okay, next.
Whole question?
Yeah?
Yeah, I think we're.
Yes. Now remember, there's more than one answer here.
Right, you can pick multiple choices here.
Sorry, I couldn't get that off my screen unless I escaped the presentation.
Oh.
Yeah, so you can pick more than one answer. All right, looks like we're about ready. Here we go.
Yeah, so it's all but the last one, Canada. What I didn't mention is on the travel history. Some countries they can't have been, like the UK. You can't have been there within the last year. But Canada's okay. So as a new donor, again, what we do require, as I mentioned before, is that in order to use that term, qualify as a donor, we call them qualified donors, you have to have that 2 medical examinations, 2 separate health screenings, and 2 sets of infectious disease screening tests before we'll use any of that plasma. Again, if you donated and didn't like the experience, even though everything tested negative, it would never get used in making any of these therapeutic proteins. Okay. So the steps, as we do as you come in the door, is they verify your ID again. So you have a government issued photo ID, can be your social security number or your tax identification number. You have to have also a suitable address within a certain area in and around that donor center. It has to be a permanent address, not a shelter, or not even the Ritz-Carlton doesn't work as a permanent address. So again, we look at addresses, and they have a listing in each center of addresses that would not be appropriate. We also then check each and every donor, and each and every time we check them against the National Donor Deferral Registry. And again, this is a nationwide database that has deferred people from giving blood and donations because of testing positive. So even if it was a false negative, once you're on that National Donor Deferral Registry, you stay on that. And again, if the center is located around other plasma collection centers, remembering I told you that the donor can donate every 72 hours, so twice a week, but not more than that because it's not safe for the donor. So what we do, if in the case of other centers being around, because this is a paid donation, and we'll talk more about that in a minute, but some people sometimes will donate more than they should. So each center in a geographic location will have a finger die and a finger designated for that center. So say the Grifols centers had the pinky. So when that person comes in to donate, they put their hand under the black light. And if they've donated that week for Grifols, we'll see the die will illuminate under the black light. Or if they've donated somewhere else, we'll see another finger. That everybody uses this die method. And then we'll check with the other center to make sure that they haven't donated more than they should have, because it's just not healthy for them. Next. So again, the medical assessments are conducted by trained and certified members of the Grifols Medical Operations Team. The centers are managed and there's oversight from a physician for several centers, and that physician manages all of the training for those people that are doing the medical operations. So that person will have a comprehensive physical exam, and that happens for the first 2 donations, and then it's an annual process. We do body mapping. We're looking for tattoos or piercings, which you can't have had within the last year. Each and every time, though, we'll monitor vital signs. We'll look at your blood pressure, your pulse, and your temperature. And now, especially as you come in, we're checking your temperature with looking at for a COVID clearance. Weight, you have to weigh at least 110 pounds. And we monitor any donor weight fluctuations that are not intentional. We look at red blood cell volume. We look at protein levels. So we're looking at making sure that your red cells are where they should be, your liver function tests are where they should be, and overall general health. We also make sure that we're educating the donors so they know really what's happening with the therapy, the plasma that they're donating, and again, the importance of having that plasma and the uses that we have for plasma therapies. And we know that a lot of our centers will participate in the walks and take a very active interest in the patients that their plasma is serving. So in addition to those comprehensive physical exams, the plasma donors have to have that questionnaire that I was telling you about. So we're looking at travel history here. We're looking at medications. This would be the same type questionnaire that you see when you give whole blood. So we look at medical history, risk assessments, any illnesses or symptoms, and social history. And some of our centers will do drug screening also. Next. So the process, as we said before, can be anywhere from 60 to 90 minutes. And again, the donors are very closely monitored. They have one phlebotomist dedicated to them, and they are one-on-one within that donation area. And we have to have that 2 times donation before we use the plasma because of the inventory hold. The donation can be no more than two times in a 7-day period. And the donors, as I said, are compensated for their time and their commitment to the process. We're not buying the plasma. Again, this is a donation that compensates them for the time that they are spending within the center. And that varies with location, and sometimes the centers will have promotional type bonuses that they'll do with the people that are donating. And I will tell you that the donors are very loyal to come and donate, and we really appreciate that. In the number of centers that I have visited, talked to donors that have been donating for well over 10 years. And again, they're very committed to it. So the donor center medical directors are responsible for all the medical oversight and the training and the certification, and they have to do yearly certification. And again, they have to be compliant with the procedures and the quality assurance standards. And each plasma center has anywhere from 25 to 50 fully trained staff personnel that perform these critical functions. Next. So we have issues again that we have to worry about with transportation and logistics. So remember, I told you that within a half an hour of the donation, the quart of plasma that they collect goes into the freezer. So that if they take a sample off and that goes off to the lab for testing, the plasma then for that inventory hold goes in the frozen state in these refrigerated trucks to the warehouses in California, North Carolina. And again, they're temperature controlled and monitored throughout the trip. We can't have them defrosting and refreezing. And again, they're in that inventory hold until the samples are analyzed and the plasma is released for production. And again, to make the medicines that we need to make from our plasma. The warehouses are enormous. So you can see from no, you can go to the next.
Okay.
Yeah, so you can see from this picture that they are racked. The donations are racked and they're all dated. So we know that what's being held for 60 days. It's also they're recorded so that if we had a change in the health of the donor, we could go and pull and discard the previous donations. So they'll take these containers, and when they're about to pull them, they go through a cutting machine, which slices off the top, and the plasma then is pooled. Next. So in the testing, when we do the testing, again, we're looking within those testing centers. They do extensive quality control and safety testing. We have, as I said before, over 250 donor centers, and every day, Grifols collects somewhere in the range of 25,000 plasma donations across the United States. So that testing center is getting anywhere from approximately 41,000 samples per day to test. The donation testing is done on each and every donation. It's then we do the testing on each and every lot that we are making, and we save a sample of each and every lot indefinitely. So if we ever had to go and do a look back, we would have the lot and the sample. So they're doing 87 million tests a year. Again, these are the two labs in San Marcos and Austin, Texas. Okay, next. So each and every test is what we are doing here. These are state-of-the-art labs. They're amazing to see. Each and every donation is tested for HIV, hepatitis A, hepatitis B, hepatitis C, and parvo B19. And then we do testing for the donor. We're doing periodic testing for liver function tests. We're looking at CBCs, we're looking at blood cell antibodies, and we're looking at syphilis. I will tell you, this is a perfect time to mention this, that Grifols is also doing, looking at collecting and developing a hyperimmune for the COVID virus. So we're looking at the antibodies and looking at people that have recovered from the virus. So we can, in fact, make a super immune globulin with multiple antibodies to use as we have a vaccine, as a prophylaxis for people who have had COVID, or people to prevent COVID from happening going forward. And that process is starting. The manufacturing process will start in May in Clayton. We did the same thing in Africa for Ebola, which was very successful. And we are going to start that process in May, now depending on the collection of and the people who have recovered to shore up these antibodies to make this hyperimmune. So next. So now, once we have it pooled, even though everything has tested negative, the FDA requires that we do at least 2 viral inactivation and removal steps. And Grifols goes above and beyond that and does multiple processes depending on the protein that we're pulling. So for alpha-8 and alpha-9, our clotting factors, we do pathogen inactivation with solvent detergent, heat treatment, and lyophilization, which means we're making it into powder. And then pathogen removal. Pathogen is essentially a disease-causing agent, a word for disease-causing agent. We do precipitation, chromatography, and nanofiltration. So we're going to go through that. I think we have the next question. Don't we have one in here? Oh, we have, okay. Yeah, see if people know this. Okay. Right, everybody got it.
I think we have one more question.
I think it's after the next slide. Is it?
I thought I had them both back to back. Are they both back to back, Madonna?
Okay. Good time for the next slide. So for those people who weren't clear, so solvent detergent, our viruses are either what we call lipid enveloped or non-lipid coated viruses. And what it is a fatty substance that the virus has that's enveloping the virus and essentially protecting it. And what the solvent detergent does is break down that fatty or that lipid coating so that we can destroy the virus inside. So the COVID virus is in fact a lipid envelope virus, and we could destroy it. Keeping in mind too that COVID is not a bloodborne virus, so it's not going to be in this. Oops, did somebody ask a question up there?
Yes, so Kevin asked the question, with the current precautions with social distancing your plasma collection centers are following, are you seeing a greatly reduced volume of plasma collected? And if so, will this affect the amount of factor products that can be produced?
Actually, we are not seeing, again, what we are doing is protecting both our donors. With the social distancing, we're not scheduling as many new donors, but our existing donors, what we're doing is scheduling them so that there's not as many people within the building at the same time. So we're spacing the appointments also. Just the new donors we haven't, we are, again, limiting the numbers to be able to provide that social distancing.
So Kevin thanked you for that. And I think we have another poll question, our last one.
Okay, we can go to the next slide.
Oh, did you want to answer that?
There you go.
Oh, here, I thought, yeah. So everybody got that right. So heat treatment, again, heat treatment, we know, again, this community is very familiar, I think, with viral inactivation based on what happened prior to 1985. But had we known about that HIV was a bloodborne pathogen, heat treatment would have destroyed it. Because we know that heat treatment, which is again once the product is lyophilized, made into a powder, it's put in a water bath in the vial in heat treatment. So it stays at 80 degrees centigrade for 72 hours. And what that does is inactivates both the lipid enveloped and the non-lipid enveloped viruses. So again, that's lyophilization, dries the product and has the capacity to inactivate some of the viruses. So what happens to, again, we have all of these steps that are built in. Remember, the FDA only requires 2, but we have all these steps built in. And what's happening here is that with our factor VIII, now the plasma goes in after it's been virally inactivated with the solvent detergent. It goes into a centrifuge. And that little diagram that you see in the middle is, you'll see the little, almost look like little prongs on the side. The centrifuge is spinning, and we have what we call a ligand on the outside. So it's a heparin-based ligand, and it's like a magnet. It's pulling the factor VIII and the von Willebrand factor to the side, and the other proteins are washing through. And it says here, proteins and viruses. Were there any virus fragments, they would wash through here too, in case they were present in the solution. But this is pulling the von Willebrand, the factor VIII, off to the side, this precipitation process. Nanofiltration, we're able to, again, filter some of the proteins depending on their size. Like factor VIII can't be filtered, factor IX can be filtered. Again, if there were pathogens in the process, this would pull out any of their fragments. And again, the filtration we do in a number of our proteins, depending again on the size. So next. So the question always comes up about, what about the next virus? The one that was the one that came to mind prior to COVID is first they establish whether or not the virus is a bloodborne virus. And then they'll do experiments where they are taking a plasma sample and spiking it with a known amount of virus, either a sample virus or a model virus. And they'll put it through the manufacturing process, like obviously separate from where the products are manufactured. But they'll look at that and to see whether or not those processes were able to remove the virus that they've put into the plasma to make this a safe product. And a good example of that one would be what we did, next slide, with variant CJD. So variant CJD, again, is a prion. And again, it's a serious disease that results in the degeneration of the neurons in the brain, and it causes a progressive dementia. And it's a fairly rapid disease, which is it's rare, but it's a fatal disease. Next slide. So we did that. Once we started to see that, and again, this was where the travel warning came from in the UK. It was, you'll probably recognize the name when I say it was the mad cow disease. And again, what we did was put it through that process, and Grifols again was able to generate the data and the clearance of this prion and in the manufacturing process. And the FDA then allowed us to even put in our package insert that the steps that we use with viral inactivation and removal would likely remove the variant CJD virus, prion. So next. And again, removing it from the starting material. So we are constantly watching what's going on in the world, as we know with this particular virus, that you can go from point A to point B, and these things spread very, very quickly. But Zika was the one that we watched very closely, and we were able to offer a lot of assistance in establishing a test for the Zika virus too. So we have close working relationships, besides our being a global companies that we have sites throughout the world. We do careful monitoring and scrutiny of all the scientific publications and the new emerging pathogens that are hitting us. So we work very closely with the CDC, the FDA, and the EMEA in Europe. And this is what we're going to be doing with our convalescent plasma as far as COVID goes, working with these organizations to hopefully be able to provide something that's going to be helpful. So the whole point in developing these hyperimmunes is to provide the antibodies which allows somebody to have active antibody against the COVID virus. This is the theory that these products are going to work, because when you get a vaccination, there's a period of time that it takes your body to develop its own antibodies. So this is what we call passive immunity. We do a very similar type thing for rabies prophylaxis to allow your body to have some active antibodies while it's waiting to produce its own. So we collect a lot of plasma from people who have recovered to have this antibody pool, again, to be able to provide this therapy as we're giving people vaccination, again, to be able to start their own antibody production. So I believe that was the last.
Yeah.
Yeah.
So does anyone have questions? Feel free to unmute yourself if you have a question.
And if you have any questions about whether you know somebody that has recovered from COVID and would be interested in donating their plasma, we can certainly provide you with the numbers to be able to have them know where to go as far as the center goes.
Madonna, you want me to stop? Oh, I think you have some questions up there, or two orange.
I haven't seen any more questions. So you can, yeah, if you want to stop sharing, that works.
Okay.
I had a quick question. So we have several collection sites in Oregon. We were supposed to do a live, visit to one of those. Are there any extra precautions that you take in certain parts of the country that are different from other parts, or are they all almost exactly the same?
As far as the collection, well, when we do look at people's travel history for COVID, we look at whether they've been out of the country or have they traveled from a particular hotspot.
Ah,
So
yeah,
we'll look,
no.
yeah. And I live in the Boston area, and we're having a hotspot.
Yeah.
Yeah.
Good to know. And then the other question I have is, if someone wanted to work in one of those collection sites, what kind of a degree or background would they have to have?
It varies because there's different roles. So from phlebotomy to the medical personnel to administrative people, lab people. I mean, it varies depending on what the job would be, but we have a very long training process. So they would go through the Grifols Academy in Glendale, Arizona, and they have to certify, and they have periodic licensing depending on their certification within Grifols. So we are very, I think, very zealous about having people that are very qualified to do what they do. And they go through an internship, so they'll be in what they are.
Other questions, anyone? So I've been really fortunate. I got to go to the plant in Southern California and watch them actually put it through the process and the filtration and the solvent. It was fascinating. I have to say, the one thing that was the most fascinating to me was there was the clean side of the bench and the dirty side of the bench, and you felt like all I did was just cross over the bench and I went from dirty to clean. But that was really fascinating. And the other thing is, when we did the live tour a couple of years ago, we got to go into the coolers, and they were, I don't even remember the temperature, but it was like degrees below zero, like incredibly cold, and quite fascinating to go in, and they let us go in and run around in there and come right back out. But it was pretty cool.
And they show you where they have the things that are a new donor, and they, a single donor. It just goes on and on where they're placed in the freezer. Yeah.
Yeah, and they talked about the machines and how it splits the blood and puts the red blood cells
Right.
back in, and then pulls the plasma out. It's just fascinating stuff. So hopefully we'll get a chance to do a live tour again. I think that to me is the most fascinating part of all of it. Virginia, you did a great job explaining it, but I really hope there's
Yeah, what I didn't say, and I think it's a good time to say that when you're hooked up to that machine, it's a single use set. No body fluids touch the machine itself. It's a snap-in cassette, which separates the plasma from the blood cells, and then it's discarded. It's a single use device.
Kevin, did you have something, Kevin?
Oh, yeah. I did a live tour of the one of the plasma centers up in Washington. It was fascinating.
Isn't that great?
Yeah, and another thing that I really was amazed by, that each vial of Factor product has a code that can be traced back to its donor that is in that vial. So it's
wow.
Yeah, that's what we didn't go through the slides. We have another program at the very end of the slide thing that talks about Pedigree Online.
Wow.
Yeah.
So Pedigree, we have a process where you can go and look at each and every donation that went into that vial with your healthcare provider. And then we do laser etch each and every vial. So if we had to go look, the manufacturing process is filmed. We had to go look at a vial, we could find it in the filming. And then we put holographic seals on the boxes so they can't be tampered with too.
Yep.
So it's, yeah.
Yeah, I have a collection center, maybe a 5-minute walk from my house.
Oh, do you?
And back when I was using a Grifols product for a while, it was neat to think that, the medicine in the vial could have come from down the street.
It could have. It very well could have. But and our donors are. Years ago, we took a bunch of. We actually put a contest in the centers. The Immune Deficiency Foundation was having their annual meeting, and we had a contest for donors to win a trip to go to the meeting. So we had 3 girls win. They just happened to be young girls, but they all had donated for at least 10 years. They got there and they were in the booth, and the patients were hugging them, thanking them, because it's life-saving therapy, and then they got up on stage with Dr. Rosa Bray, who's the medical director for all of the centers, and they got a standing ovation.
Aww.
They were crying. I mean, it was so touching to see. They were so excited, and they love knowing that what they do is not just for the amount of money they make when they donate, but they're there for a good reason, and they love knowing the patients and seeing the patients. Yeah.
Virginia, since you mentioned that, do you want to share with everyone how many donations it takes to treat a hemophiliac for 1 year?
I think it's how many donations? I think you know better than I do. 120, is it? Yeah.
Or I thought it was 1,200.
1,200, yeah, I was close. I missed a 0.
Yeah.
Yeah, 1,200, yeah, and with the other products too, particularly with an immune globulin, it's even more than that. Yeah.
Yeah.
So the other thing that people should know, a lot of companies have plasma-based products, but I think Grifols is the industry standard, and they sort of set the standard for everyone else to meet. And so if you are using some sort of a plasma product, you can be rest assured that Grifols sort of set that standard for you, and that a lot of the companies follow much of the same requirements and sort of safety that Grifols does. But certainly Grifols is definitely the industry standard.
We were a company that is founded in Barcelona. They now have found some documents that say over 100 years ago, but the active up and lab portion was started in 1940, and they developed the whole process of plasmapheresis.
Wow.
Yeah,
So
saved a lot of people's lives during the war.
Yes, and they were isolated. They were cut off with Franco, so they had to make all of their own equipment. It's really fascinating to go and see
Still,
the.
still family run business.
Yes.
Yeah,
Well, it's a publicly traded company now, but it's still,
yeah, family
Yeah.
run.
because I'll have people say, "What's a Griffle?" It's a family name.
Kevin, did you have something that you wanted to say?
Yeah. I had a sciencey question.
Mhm.
For the plasma, does it matter what blood type it is, or is all blood types?
No, because there's no red cells.
Yeah, so
that
the
doesn't matter.
blood
No.
type doesn't matter. That's what I thought. I just
Right. Right.
Yeah, so hopefully we will get another live tour. I'd like to get that back into the schedule at some point because I think a lot of people appreciate that. And for some of you, you might know them, but we actually have someone in the community that works at one of the plasma collection sites. So it's kind of fun to ask them some questions occasionally as well. So if you're interested, I can connect you with that individual. It's a pretty awesome job.
Do you know what they do there?
No, I don't actually. I should find that out, shouldn't I? I should have asked that before tonight, but I do know that we have an affected individual that works in one of the plasma collections, so it's kind of cool.
Yeah. They're very proud to share what they do because they do such a great job.
Sure. And when we get a tour of it, when we did the live tour, it was pretty awesome that the people that were donating, we could actually say, you know, what you're donating could potentially help one of the individuals that are on this tour. I know when we first adopted Gavin, he was on a plasma-based product because he had not had more than 20 doses in his lifetime. And so we were on a Grifols product when he first came into our family a couple of years ago. And so it was rest assured for sure for us that Grifols does as much as they do. It really helped us feel so much better about the choices we were making for his treatment.
Yeah, that's great. That's great to hear. Is he watching?
Yeah, he's in the background. I can see. It's a little above his head, but that's okay. So does anyone else have any questions? Feel free to unmute yourself and ask questions or share anything.
And I would encourage you, if you know somebody who's recovered from COVID, that you encourage them to think about donating their plasma.
That would be great. Absolutely. Well, thank you so much, Grifols. Thank you to Colleen and Virginia. I really enjoyed it. I've even been to the collection site and I learned a lot even through this tonight. So thank you so much. Everyone else, I hope I see you again on another Thursdays at 7, especially the campfire next week. Otherwise, Tuesdays at 2. We'll see you then. Everyone have a great weekend. Thank you.
Stay well.